Chapter 6, Section 9, Translation of the Genetic Code. Okay, remember, comes to the messenger RNA, everything is read. 3 by 3 by 3, codon by codon by codon. 64 possible codons, okay? 4 nucleotides, but you have 3 possibilities, so that's 3 to the 4th. 64 possible permutations. When you go through for each permutation, each one of them calls for a different amino acid. Kind of cool. Unfortunately, though, there are, 3 of them, UAA, UAG, UGA, that do not call for any amino acid. These are the stop codons that tell the ribosome stop, terminate translation, fall off. When it comes to prokaryotes, or basically all living things, AUG is the start codon. Just turns out in bacteria, AUG, the first initial AUG, is for N-formal methionine. Turns out all the proteins begin with this F-met. They call it F-met, formal methionine. It may be cleaved off later, post-translational modification, but all bacterial proteins start with this. All of ours start with methionine, which will be cut off the majority of the time in post-translational modifications. They call this a degenerate code, okay, because all 64 are present, but there's only 20 amino acids. Some of them are going to have more than one codon that can call for it. But each codon can only call for one amino acid. So, what's interesting is if you go back and you look at that table, your UA, CCG, CCA, it's either going to call for the same amino acid, or if it's calling for 2 amino acids, that one change at the 3rd nucleotide, the amino acids that are different are still going to be closely related. They're still going to be acidic, or they're both going to be basic, or their shape's going to be close enough that it's not going to interfere with the structure. They call this wobble. You have to have the first nucleotide in the codon exact. Second one, yeah, not quite. Third, there's wiggle room. Still get this either the same amino acid or one that closely resembles it. It's kind of a redundancy that hundreds of millions of years of evolution has given us. First one's got to be spot on, second one to a lesser extent, third one, you got some wiggle room, hence the name wobble. It's interesting, though, that even though there's multiple codons for some of the amino acids, they're not all used equally. There's going to be one that's more preferential than the others. Don't know why. I'm sure somebody's sat down and figured this out or has some philosophical reason to explain it. I don't know. It's going to be different organism to organism, species to species. And a lot of it's going to correlate to, what tRNAs are available in that cell, in that organism. Is there one tRNA with the anticodon that seems to be more predominant than the others that are also for the same amino acid? As I said, start codon, formal methionine, also known as F-met, just straight up methionine and archaea and eukaryotes. Reading frame, for all living things, it will be triplet, 3 by 3 by 3. There's these things referred to as the Shine-Dalgarno sequences, or you'll see it nowadays referred more to as ribosome binding sites, RBS. These are going to be sites that are right before the beginning of, codon number 1, that are going to be where the ribosomes bind begin the interaction, and then slide down to codon 1. Stop codons, well, they're pretty much the same for all living things, UAA, UAG, UGA. You'll see in some organisms, very few, selenocysteine, pyrrolysine, these are variant amino acids, may override the stop here. For one of these stop codons may code for these in very few organisms under certain conditions. And it's very important that everything gets positioned correctly, because I mean here this, very basic figure, but it really shows you what's happening. The correct reading frame, ACA, is where you start. ACA, UAC, blah, blah, blah. Threonine, tyrosine, arginine, serine. You start one ahead. Aspartame, isoleucine, proline, isoleucine, completely wrong amino acids. You start one behind. Histidine, threonine, aspartame, histidine. These two won't make a working protein. They'll make a polypeptide that has no shape, thus no function.